Now, we can finish our talk on proteins, talking about protein folding, how protein folding is achieved, and talk about chaperones. Now, given that the shape of a protein dictates its function, the ability of a protein to fold into the right shape is very, very important to making sure that it works properly. And it was once thought that every newly made protein could find its correct conformation, its correct folding pattern on its own. Now, there are cases where you can take a protein in vitro outside the cell and denature it and break its tertiary structure, break all hydrogen bonds that might be holding it together, any ionic bonds, and those proteins can refold into a native state, into a correct conformation, in other words, a correct three-dimensional structure, a correct tertiary structure. But we now know that chaperone proteins help proteins find their correct conformations. They help fold proteins. And there are known diseases where deficiencies in chaperone proteins cause the disease. Cystic fibrosis, for example. In some cystic fibrosis individuals, the protein that is mutated in other cystic fibrosis patients appears to have the correct amino acid sequence. It doesn't appear to be mutant, like in other cases of cystic fibrosis, but it fails to fold, presumably because of mutations in a chaperone protein. And the chaperone proteins assemble into large complexes that help protein folds. This is very schematic, a very schematic view of this. There's binding sites for proteins, ATP is required, and correctly folded proteins come out of the process. But a lot of folding of proteins can occur spontaneously, and I'll talk about that in a second. Now, proteins once folded, can be denatured by a variety of conditions. For example, pH changes will affect the ability of a protein to maintain its correct conformation, its correct three-dimensional shape. And you can imagine that pH changes change the concentration of hydrogen ions, which can affect the protonation state of amino acid side chains. And if, for example, hydrogen ion concentrations are raised, previously negatively charged acidic side chains might be protonated, might take a hydrogen ion and be neutral. And if that acidic side chain of that amino acid was important for a salt bridge, for an ionic bond, in other words, if that were the case, then that would be disruptive. So you can imagine, for example, in this protein here, let's imagine that there's a side chain of one of the amino acids on this alpha helix and a side chain off this stretch of protein here. Let's say that this is a positively charged basic side chain, and this is a negatively charged acidic side chain. That's an ionic bond. And in proteins, we often call ionic bonds salt bridges, a salt bridge. And salt bridges can help stabilize protein structure. Well, now, if you change the hydrogen ion concentration, that can affect whether or not this basic amino acid remains positively charged or whether this negatively charged acidic amino acid remains negatively charged. And therefore, you can break an ionic bond by changes in pH. Temperature, of course, increasing the kinetic energy of the molecules in solution will bombard the protein with high energy moving molecules. So as you increase the kinetic energy in solution, that the kinetic energy of molecules bombarding the protein is increased, and that can break some of the hydrogen bonds and weak bonds that hold the conformation in its three-dimensional shape. So the protein will go into what we call a denatured state, where it's not correctly folded anymore. And the ionic concentration of the solution obviously can be very important. If you throw in a lot of positive ions, for example, a lot of salt, a lot of sodium ions, a lot of chloride ions. Those could neutralize charges on polar amino acids and thereby disrupt the role that those polar amino acids play in maintaining a correct three-dimensional shape. So denaturation can occur. So let's look at case of protein folding spontaneously. And I'm drawing the main chain with a line and then side chains coming off of different amino acids with these little lines. And let's designate X as being hydrophobic side chains. Oops, that's it. Hydrophobic side chains. And let's say Y equals hydrophilic, water-loving hydrophilic side chains. That is polar side chains, or charged side chains. So hydrophilic, polar, or charged, or X is hydrophobic. So let's imagine that we have, say, an X here, and X here, and X here. These are hydrophobic. And we'll say Y, 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 Y. Let's imagine. There it will be considerable thermodynamic force forcing the hydrophobic side chains into the interior part of a protein so that they can hide away from the aqueous water environment. You'll remember early on we talked about the ability of water to organize nonpolar hydrophobic molecules. Well, this is a good case of that. The protein would fold so as to shield the X's, the hydrophobic side chains of hydrophobic amino acids, away from water, just like oil is partitioned in water. And the hydrophilic side chains of hydrophilic amino acids, on the other hand, would mix with water because it's attracted to water. They're polar, so and water is a polar solvent. So the hydrophobic effect and the polarity or lack thereof then can help fold a protein into its correct shape, independent of chaperone proteins. Now we're going to end this lecture here, and next week actually we'll be a little behind, but we're going to tackle lipids next week.